The optimal sequencing really depends, in my opinion, on the genomics that the patients present with. Ideally, for patients that present only with the MYD88 mutation, BTK inhibitors represent the best first-line option for them because we see very high rates of activity, even long-term durability. In fact, in our Ibrutinib monotherapy study, 70% of the patients who had previously received therapy with another agent had in fact remained progression-free at 5 years. The data from our frontline trial even looks more exciting. In fact, we're looking at around 92% progression-free survival at 4 years. So we know that these drugs work well. But if the patient has the second mutation, the CXCR4 mutation, which we see in about 40% of patients, that treatment duration will be shorter, and it will take longer to get patients into deep responses. And so here we have to be very careful as clinicians to try to understand what exactly the needs are of the patient. If they're presenting with hyperviscosity crisis and they have very high IgM and you need to get that down, maybe a BTK inhibitor isn't necessarily the right answer for that patient because it'll take a long time to get there. And this is where maybe bendamustine or proteasome inhibitor-based therapy may be necessary. But there are also MYD88 and CXCR4 mutated patients who don't need that immediate response, where a BTK inhibitor may in fact be the right choice. And then there's that small population of patients, the ones that have no MYD88 mutation. We call them MYD88 wild-type patients. Their genomics are very different. They tend to parallel very much those that we see in DLBCL. And a drug like bendamustine or proteasome inhibitor-based therapy might be appropriate, but we're now also seeing that Zanubrutinib, a new BTK inhibitor, also is showing activity in this population of patients. It may be, another option for patients who don't have the MYD88 mutation.